Okay, it's Mrs. Shannon. We're going to talk a little bit of evolution and how it relates to matter processing in cells by looking at three domains. And so this kind of goes in with the origin of life and the evolution of the first cells here. And so in the Bozeman video, I talked a little bit about this. So please use those Bozeman videos. But the big questions that this is going to answer is how can we think about organizing the diversity of life? And so the life that we know now is very diverse, different functions, different structures, etc. And then we need to look at these different organisms, these different forms of life, and find major features of organisms that we can use to classify them. What do they share in common? And these structures and features and behaviors that are shared oftentimes suggest evidence for common ancestors. And so we're going to look at a couple of major ones here. And so before we begin, we need to understand that our understanding of life's diversity is constantly expanding. It's expanding as new organisms are found. It's expanding as new lines of evidence are uncovered or put together. And so this presentation reflects a very brief explanation of some of the relationships among organisms. It's not complete. The branches of the tree only represent general relationships, not time, abundance. Don't use it as a measuring stick. And focus on the big picture, the hows, instead of the nitty-gritty details. Don't focus on what these structures or forms are, but how they arose and maybe why they arose. Like, what did it enable? What was the positive here? And so first of all, we have this division. We have a division between eukaryotes and prokaryotes. And so that's a major cellular division. Prokaryotes are before the center, or karyote is sometimes referred to as the nut or the egg. There's no nucleus or membrane-bound organelle. And two of the three major domains of life include prokaryotes. Now, we talk about domains. We used to talk about kingdoms. Domains are above kingdoms now. So when I was in school, it was just kingdom, phylum, class, order, family, genus, species. Now we have three major domains that's just above the kingdom level in classification. Eukaryotes have this true center. They have a nucleus and they have membrane-bound organelles. They have a true nucleus. The third domain of life contains eukaryotes and the 4 out of 5 of the classic kingdoms. And so when you're looking at a classification chart or you're looking at a tree of organisms, keep in mind that differentiation of terms, that all 4 of the 5 kingdoms are found in one domain. So the 3 domains diverge very soon after life's last universal common ancestor. And what that means is this first common ancestor is where those biomolecules started forming and having a sense of order and were able to function and process and interact and be able to be process matter and process energy, and it's able to pass on information. As soon as that last common ancestor was formed, we had this divergence. So here we have bacteria, archaea, and eukarya. And so here, bacteria, we have our common ancestor. Okay. We have archaea, the ancient organisms, which also diverged eukarya. And then we have the bacteria. So we have our last common ancestor right around here. There's a really cool later on in the semester, or in the year, next semester, we'll look at a New York Times recent publication of the most recent thought of the tree of life, and it looks more circular like this than it does in the actual tree form that we're used to seeing it in. But here you can see the universal ancestor and the more classic tree look to it. And you can see the eukarya, the archaea, and the bacteria. Most of our focus in AP biology are on these organisms. There is a chart that has some of these common features of each of them. You don't need to memorize this chart. This is the what that it was saying. Don't get bogged down in the nitty-gritty, but instead start opening up to the possibility and the idea of how these things came about or when they came about and what circumstances were there in the environment to support the characteristic to flourish. So the universal common ancestor, we're not sure what it looked like, but we can make some inferences. And this is just based on kind of like Occam's razor and the evidence that we have. Occam's razor, if I don't know if you ever learned it in like physics or chemistry, it's like go with the simplest explanation for the evidence that you have. And the evidence that we have support that it's unicellular. It's not complex. Needed to be able to do all of its functions within one cell. It's heterotrophic. It couldn't produce its own energy. It had to gain it from other materials around it. That production of energy from the sun, something that came later. It's more complicated. It needed organelles to be able to do that. And then DNA as genetic information. And so we've moved past this RNA idea. So as the life was being formed, as these biomolecules were coming together and make protocells, these before cells, RNA was probably the carrier. But these were in the protocells before it could really function and complete all of life's characteristics on its own. Once you had this, I want to say organism, but don't think of it as complex. Once you had this living form that was able to process matter, process energy, transmit information, reproduce, and I'm missing the last trait here, pass on its information, and communicate with others. At this point, it was probably DNA as genetic information, and that is just because of stability purposes of the versatility of DNA. So when we're looking at bacteria, general characteristics of it are that they are prokaryotic, unicellular. They have various nutritional modes, whether it's from other chemicals, whether it's sulfur vents that it feeds from, whether it's organic matter, etc. So here's an anatomy of a bacterium. It's made of a peptidoglycan. Oftentimes they will have a flagella to help it move around. The pili are like sticky substances, kind of like cilia, but more so to help it stick to things. And so it really uses its other organisms to kind of help pull it along. The different types of bacteria are separated by their shape. And the different types of cell wall actually help differentiate whether it's gram-positive or gram-negative. It's a way to tell what the cell wall is actually made out of. So when you hear gram-positive bacteria and gram-negative, that's what it's referring to as a type of cell wall. The bacterial cell membrane is the major organelle used in bacterial metabolism. So it's different than a eukaryotic cell, which uses these other organelles. Instead, they use their membrane. Bacteria have evolved all major forms of metabolism, so independently out of away from eukaryotic cells. They have photosynthetic prokaryotes, so using sun energy, and we have aerobic prokaryotes using oxygen. Okay, we'll spend some time looking at genetics, bacterial genetics later on, so I am actually skipping this part. When we look at genetics, we'll come back to this domain. They have some unique properties of being able to divide by fission. Their genetic information is in circular DNA. And the way that they reproduce and transfer is through transformation and conjugation. And we're going to just go through that. So when we do genetics, we'll come back to it. Bacterial ecology, we have some useful examples. First of all, bacteria live everywhere. There's more bacteria on you than body cells. There's just that many. They tolerate vast ranges of environmental conditions, and they have a wide diversity of nutritional modes. Some are good, some are bad. Usually they get the bad rap for causing illnesses, but they also help in protection of the organism's body. They help in breaking substances down. And so these bacteria help absorb soil nutrients. So like if you're looking at nitrogen fixation, we have some that live in marine organisms providing bioluminescence, and so they have this sort of symbiotic relationship. And so they have all these little niches that they fill in the environment that allow them to remain. Now, of course, they are pathogens as well, but it's a very small number that are used to cause disease. Here's an example of some of the more popular ones. And this is often why they have the bad rap here. We have the deer tick that contains a bacteria that spreads Lyme disease. Now, bacterial infections can be treated with antibiotics. We'll look more at this when we look at natural selection and we look at evolution. Antibiotic penicillin was an accidental discovery in 1928, but again, many of these bacterial infections can be treated with antibiotics. Now, I want to also talk to you a little bit about archaea. It's the domain of the weirdos. It was discovered in the 1970s, and it was thought to be bacteria, and so it was actually called archaebacteria up until the 1990s. And so I still, because I went to school in the 90s, have a problem. I call it archaebacteria, even though it was just kind of its odd man out. No one knew much about it. This is like the ancient organisms, ancient life forms, but they have enough unique properties and combinations of the other two domains to qualify as their own. So they're not really bacteria. They're not really eukaryotes. They're sort of a little bit of each of them and a little bit of their own. So like bacteria, they're prokaryotic, unicellular, various nutritional modes, but they also live in extreme environments. And so when we say extreme environments, we're talking about like deep sea ocean vents. Oh, and here, this is kind of cool here. You can actually see the comparison here. And so here we have archaea in the middle, and this is what we had before, but like circular chromosome, just like bacteria. Growth not inhibited by antibiotics, just like eukarya. And so we have the same initiator amino acid. So when we talk about the start codons and stuff, methionine for both archaea and eukarya, but it's different for bacteria. And so it's sort of its own hybrid category. Yellowstone's hot springs contain archaea, acidic mining runoff, your water heater, all extreme environments. Methanogenic archaea are able to convert complex hydrocarbons into methane. And so when we're looking at different forms of clean energy, that's actually a biotech field that's specializing in using bacteria to help in this clean energy process. And then we have eukarya. So when we're talking about cells and cell structure and we're talking about systems, most of the time, 90% of the course, we're going to be focusing on us, okay? Animals, fungi, plants, and protists. Those are the four kingdoms here in the eukarya. And so most of our AP biology is going to be focused up here. Nothing really on the archaea and a little bit on the bacteria, especially when it comes to genetics and how we can use bacteria in biotechnology. So we have protists, plants, fungi, and animals. And so when we're talking about these different types of organisms, prokaryotes and eukaryotes, and when we're talking about archaea and bacteria and eukaryotic cells, it's nice to start off at the beginning of the year with this whole idea of how it sort of breaks down and where the separation is. So by the end of this, you want to make sure that you can determine if a particular organism is eukaryotic or prokaryotic. And the easiest way is by those cell organelles. If it has a nucleus and true organelles, it's eukaryotic. And if not, it's a prokaryote. You want to identify the common structures of prokaryotic cells and explain how each contribute to the overall functioning. So you want to be able to identify the flagella there that help it move, or the pilia that help it stick. You want to be able to identify the circular DNA that's within it. So those are a few of the features of prokaryotes. You want to compare the major modes of prokaryotic genetic recombination. At this point in this school year, the beginning, no, you're not there yet. We're going to talk more about this in horizontal transfer and recombination and transformation when we look at genetics and biotechnology. That way we can look at the actual application. And we want to be able to compare the biology and ecology of bacteria and archaea. And we'll talk a little bit more of ecology, especially these symbiotic relationships, when we look at ecology towards the end of the school year. So I hope this kind of helps at least giving you a clearer picture on the 3 different domains of life. And as we're going to be using them throughout the year, you can kind of have a little frame of reference of how they are interconnected. Please ask if you have any questions.